Very good morning, students. Today, let me continue with that chapter Mendelian inheritance. And in this chapter, we have already discussed about monohybrid cross, dihybrid crosses, and then some laws also. After this Mendelian inheritance, let us go through some other types of inheritance patterns, which are collectively referred to as interaction of genes. So far, you have studied that in case of pea plants, they follow the simple Mendelian inheritance patterns. But all the organisms will not follow this Mendelian principles of inheritance or the Mendelian laws of inheritance. So such kind of inheritance patterns are collectively called interaction of genes. There are different kinds of interaction of genes are there, and for your syllabus you will have to learn the inheritance patterns like incomplete dominance, epistasis, hypostasis, supplementary gene interaction, complementary gene interaction, multiple factorial interaction or the multiple gene interaction, and then multiple allelism, etc. Today let me go through this first interaction of genes, what is known as incomplete dominance. What is incomplete dominance? Dominance is nothing but a dominant gene is always dominant over a recessive gene. But in case of incomplete dominance, it is quite different, and the definition goes like this. It is defined as a process in which the dominant gene is incompletely dominant over the recessive gene and produces a phenotype which is intermediate to the parental types. It is also called as partial dominance or blended inheritance because there will be the partial dominance is going to be exhibited by the dominant gene, and it's called blended inheritance because there will be the blending of the gene influences here. That is, there will be the blending of the dominant gene influence and the blending of recessive gene influence, so that a new intermediate character is produced. Hence the concept is also called intermediate inheritance. Examples: flower color in Mirabilis jalapa, that is 4 o'clock plant, and Antirrhinum majus, which is called snapdragon plant. And in case of animals, it is found in Andalusian fowls with reference to the the color of the plumage. And in case of cattle, it is with reference to the horn nature, wherein short-horned cattle will show this kind of inheritance pattern. Let us go through one example here, which is with reference to the flower color in case of 4 o'clock plant, that is Mirabilis jalapa. In this plant, it can produce two types of pure breeding plants, that is red flowered and white flowered, wherein red flower is dominant over white flower. And if a cross is made between the two, the F1 plants do not have red or white flowers. Instead, it was pink flowers. And it is due to the interaction of both the alleles, that is dominant allele and recessive allele. So a new character is going to be produced. And the self-breeding of this F1 pink flowered plants yields a progeny of F2 generation consisting of three types of plants, that is red flowered, pink flowered and white flowered, in the ratio of 1:2:1. That is, one red flowered plant will be produced and two pink flowered plant will be produced and one white flowered plant will be produced in the F2 generation. Both phenotypic and genotypic ratios are same in incomplete dominance, that is 1:2:1. Unlike in case of Mendelian inheritance pattern, wherein the F2 generation shows phenotypic ratio of 3:1 and the genotypic ratio will be in 1:2:1 only. So this is with a cross showing that inheritance pattern called incomplete dominance, wherein the parents selected for this cross are red flowered plants, which is homozygous, and homozygous white flowered plants, and their genotypes are as follows: capital R, and white flowered plants will have smaller genotype. And the gametes produced are as follows: wherein the dominant parent produces only one gamete, one type of gamete, that is having capital R, and same with reference to the recessive parent, that is white flowered plants. And in the F1 generation, as we already discussed in previous slide, it will produce all heterozygous pink flowered plants. And when selfing or self-pollination is achieved by means of crossing the F1 progeny, the gametes produced are as follows, that is, each parent will produce two types of gametes, one with capital R and one with small r. And when these F2 generation, when these gametes are fused together randomly, it will produce the F2 generation, which is as follows, as we have already discussed in previous slide, that is one red flowered plant, two pink flowered plant, and one white flowered plant will be produced. So this will be the type of inheritance pattern seen in case of this incomplete dominance in Mirabilis jalapa. So the phenotypic ratio, as we already discussed, that is 1:2:1, as well as the genotypic ratio. Then in case of Andalusian fowls, the fowl has two pure breeding variations like black and splashed white, that is with reference to plumage color or the color of the wings, and a blue colored third variety is also found. When a pure black is crossed with white, F1 fowls are blue colored, and on breeding F1 blue fowls, F2 generation has three types of fowls, that is black, blue and white, in the ratio of 1:2:1 respectively. The black fowl has both the allele for black colors, that is capital BB. White has both allele for white colors, that is small bb, and but blue color has only one allele of the both, that is one capital B and one small b. With reference to short-horned cattle are pure breeding variation as far as their skin color is concerned, and they show two types of skin colors in pure variety form, that is white-haired skin and red-haired skin. When the two are crossed, F1 cattle have brownish colors, and the inbreeding of F1 produces two types of cattle, that is red, brown and white, in the ratio of 1:2:1. And this indicates partial dominance or incomplete dominance. So these are the few examples of this incomplete dominance, which is also called intermediate inheritance. Now let us go through this multiple allelism. You know what are alleles are? Alleles are nothing but the alternative forms of the same gene, which is occupying the same locus of the homologous pair of chromosome. But multiple alleles are quite different. The definition is as follows. When a single gene for a character occurs in more than two alternative forms occupying the same locus on homologous chromosomes, it is called multiple allele, and the phenomenon is called multiple allelism, which is also known as gene polymorphism. The examples are as follows. Blood groups in human being, coat color in rabbits, eye color in Drosophila, self-sterility gene in Nicotiana, etc. Coming to some important characteristics of multiple alleles. Number one, there are more than two forms of the same gene. All of them are mutant of the same wild allele. And out of various multiple alleles, one is dominant over others. And they exhibit dominance, recessive, incomplete dominance, and codominance, like different patterns of inheritance is observed. All the alleles show Mendelian inheritance only. And a gamete or a chromosome carries only one allele. An individual possesses only two alleles of a gene. The best example, we'll discuss it now, that is blood groups in humans. You know, human blood groups were first discovered by Karl Landsteiner of Vienna in 1901, and he is an Austrian physicist who was working on blood sample in laboratory, and he found that RBCs analyzed clumped or agglutinated, and sometimes they did not. So in some of the blood samples, he could see the clumping, and in other samples there was no clumping. And he analyzed his results and concluded that there were at least 4 different types of blood groups in human beings. They are A, B, AB, and O. And for his outstanding work on blood groups, he was awarded the Nobel Prize in the field of medicine in 1930. And in humans, blood grouping can be based on the 2 major components of the blood, namely the plasma and blood corpuscles. One of the blood corpuscles that is involved here is RBCs. The membrane of RBCs is associated with glycoproteins called antigens or isoagglutinogens, which are mainly responsible for the determination of the blood group. And the plasma is without corpuscles and clotting elements, and that is nothing but serum, and that has antibodies or isoagglutinins, which are proteins. So it is very clear now that RBCs carry antigens or isoagglutinogens, whereas plasma carries antibodies or isoagglutinins. And there will be the reaction between this antigen and antibodies that will produce the clumping or agglutination. Coming to antibodies or isoagglutinins, they are the proteins present in the serum of the blood, and they are naturally present antibodies, and they are not synthesized against any foreign antigen as a part of the immune reaction, but these are naturally present. There are 2 types of antibodies. They are antibody A and antibody B, which are specific to antigen present on the serum. Coming to antigens or isoagglutinogens, they are small chains of sugars attached to proteins of RBCs, hence they are glycoproteins. There are 2 types of antigens, antigen A and antigen B, identified based on their sugar components. So here is the picture which will show you that a person having A blood group will have A antigen on the surface membrane of the RBC, and person having B blood group will have B antigen on the surface membrane of the RBC, and person having AB blood group will have both A and B types of antigens on his RBCs, and person having O blood group do not have any antigens but have only antibodies. And based on the antigen and antibody present in the blood, 4 types of blood groups are identified. Note that there will never be an antigen and its corresponding antibody in the blood of the same person. That is, the person having blood group A will have antibody B and antigen A, and the person having blood group B will have antibody A and antigen B, and the person having blood group AB will have no antibodies but both antigens A and B, and the person having blood group O will have antibody A and B but no antigen. The blood group in man is controlled by a single gene, that is I, which stands for isoagglutinogens, or L, which stands for Landsteiner, the person who discovered it, and with the 3 alleles like IA, IB, and I0. The gene IA and IB are co-dominant, and they are expressed together without being influenced by each other. I0 is a recessive allele, hence IA is equal to IB, which is greater than I0. Greater than means IA and IB are dominant alleles over the I0 condition, that is I0 allele. So here is the table which shows the details of such antigens present and antibodies present in different types of blood groups. For example, person having A blood group will have A antigen, B antibody, and the genotypes are IA, or I0, or small I, that can also be referred to as small I by geneticists. And the percentage of such people in population is around 45%. And person having B blood group will have B antigen, A antibody, and the genotypes are IB or IB-I0, that is heterozygous condition, or IB small I. And there will be about 15% of such people having B blood group in population. So the most numerous type is A blood group. And AB person will have both antigens, as you see in this table, and no antibodies, and the genotype is heterozygous IA-IB, and there are only 3% of such people in human populations. And person having O blood group will have no antigens but both antibodies, and the genotype is I0 because there are no antigens in their blood, so it is the genotype is written as I0 or small I, and their percentage is about 37% in human population. So below is the list of the possible type of progenies produced by different types of parents having different blood groups, and the inheritance of genes IA and IB and I0. It has been studied in human population, and based on the homozygosity and heterozygosity of the parents, blood groups of the progenies will differ, which is shown here in the tabular column. So the details of that we will discuss it in next class. Coming to rhesus factor, that is Rh factor, shortly abbreviated as Rh factor. K. Landsteiner, that is same Carl Landsteiner, Philip Levin, Stenson and Weiner in 1940 discovered a proteinaceous erythrocyte antigen present in most of the humans, which is originally present in rhesus monkey. Hence it is called as rhesus factor, or shortly abbreviated as Rh factor. And based on the presence or absence, we can identify two types of individuals, that is persons having Rh factor are said to be Rh plus, and their percentage is about 85%, while the ones lacking this antigen are known as Rh negative, and their percentage is about 15%. And the gene responsible for Rh antigen is a dominant gene, that is R, and the gene for no Rh antigen is recessive gene, and that is represented by a small r. And this gene follows simple Mendelian inheritance pattern. So when blood transfusion is performed from Rh plus person to Rh negative person, the blood of Rh person becomes sensitized and develop antibodies against Rh antigen. So it is necessary to test the blood even for Rh factor also before transfusion, otherwise it will be detrimental or lethal one. Testing of Rh blood group. Rh is tested with Rh antiserum or plasma carrying Rh antibodies. A small drop of blood is mixed with a small drop of antiserum, and if agglutination occurs in case of Rh plus blood and no agglutination in Rh negative blood. The rarest blood group is AB, and the most available blood group is A plus, that is A positive. Coming to a situation with reference to Rh factor, that is Rh factor leading to a situation called erythroblastosis fetalis, which is also called hemolytic disease of the newborn. In a cross between Rh plus, that is Rh negative woman, and Rh positive man parents, all their children are Rh positive only. And in the first progeny, if fetal blood enters the maternal blood, it gets sensitized and produces antibodies against the fetal Rh antigen. By the time antibody concentration reaches sufficient strength, and the term PF development may get over and a normal child may be born. And in the successive pregnancies, if fetal blood enters maternal blood, it becomes sensitized once again and develops some more antibodies this time, and when the antibodies reach sufficient strength, they diffuse into the fetal circulation and agglutinate fetal RBCs. So there will be the massive destruction of RBCs takes place in the fetus. As a result, the fetus dies in the womb itself and a dead child is born. And this condition is due to Rh incompatibility between fetal and maternal blood, which results in a disease or a condition called erythroblastosis fetalis, because there will be a massive destruction of RBCs occur here. And it is also called hemolytic disease because the term hemolysis means RBC destruction, and as it is seen in the newborn individuals, it is called HDN, that is hemolytic disease of the newborn. And as it involves hemolysis, it is called hemolytic. And this condition is medically helped by injecting Rh negative women by an injection called anti-D injection containing anti-Rh antibodies, and this helps to prevent the antibody forming process. So thereby we can prevent the death of the subsequent pregnancies or the fetuses. So this is a brief information about erythroblastosis fetalis, a disorder or a condition caused due to Rh factor. So this is with reference to the introductory aspect of this topic.